Gregor Mendel was grew up poor in an agricultural area of Europe and entered an Augustinian monastery at age 21. After some time, he left to study science in Vienna and then returned to the monastery, eager to begin his research. In Mendel's time, it was thought that heredity had to do with a blending of attributes, like the way two colors mixed to give an intermediate color. But Mendel seemed that heredity must be based on discrete units that can be inherited, which came to be referred as genes. In gene theory, each parent has a set of genes, and these are retained intact in the offspring. Mendel worked for several decades with pea plants in the garden of his monastery to produce an impressive body of data to support the gene theory, all of which was done decades before chromosomes were observed and understood. Now, we will discuss the methods Mendel used to breed the pea plants and the conclusions he was able to derive from them. Pea plants has a number of visible traits that can vary, so results are easy. These are things like flower color, seed color and shape, pod color and shape, flower position and stem length. To do so, he first established pea lines with two different forms of a feature, such as tall versus short height. He grew these lines for generations until they were pure breeding, then bred them to each other and observed how the traits were inherited. Generations of the pea plants are short, and the offsprings are many, so data is easy to gather, and Mendel can easily control mating by removing the stamens, the male fertilizing organs that produce pollen from a particular set of plants. Then he could deliberately cross-fertilize with pollen from other plants. This means he had complete control over which plants were mating with which other plants. Mendelian inheritance refers to an inheritance pattern that follows the laws of segregation and independent assortment, in which a gene inherited from either parent segregates into gametes at an equal frequency. Mendel's developed two fundamental laws of genetics. These are the law of segregation and the law of independent assortment. According to law of segregation, only one of two gene copies present in an organism is distributed to each gamete that it makes, and the allocation of the gene copies is random. When an egg and a sperm join in fertilization, they form a new organism whose genotype consists of the alleles contained in the gametes. Each homozygous parent in the present generation produces only one kind of gamete. The heterozygous F1 offspring produces two kinds of gamete. Self-pollination of the F1 offspring produces F2 offspring with 3:1 ratio of yellow to green seeds. The law of independent assortment states that the alleles of two or more different genes get sorted into gametes independently of one another. In other words, the allele a gamete receives for a one gene does not influence the allele received for another gene. The reason for independent assortment is to see why independent assortment happens. We need to fast forward half a century and discover that genes are physically located on chromosomes. To be exact, the two copies of gene carried by an organism are located at the same spot on two chromosomes of homologous pair. Homologous chromosomes are similar but not identical, and an organism gets one member of the pair from each of its two parents. The physical basis for the law of independent assortment lies in meiosis of gamete formation. When homologous pairs line up in random orientations at the middle of the cell as they prepare to separate, we can get gametes with different combos of mom and dad homologous because the orientation of each pair is random. Non-Mendelian patterns involve genes that directly influence the outcome of an organism's traits and obey Mendel's law. Most genes in eukaryotic species follow the Mendelian pattern of inheritance. However, there are many that do not. Non-Mendelian inheritance is a general term that refers to any pattern of inheritance in which traits do not segregate in accordance with Mendel's law. These laws describe the inheritance of traits linked to single genes on chromosomes in the nucleus. Non-Mendelian genetics, some traits exhibited a kind of blending, where the offspring of organisms with two different traits didn't have one or the other form from the parents. They had something that was sort in the middle. This implies that certain alleles are not dominant over the other ones. Rather, they share rules like harmonious friends. Some traits seem to be controlled by complex inheritance patterns. We know now that traits can be controlled by more than one gene, or genetic material may pass down from parent to offspring in many different ways than what Mendel predicted with his law of segregation. Non-Mendelian inheritance patterns might just be caused by mistakes in reproduction. There are a ton of different processes all happening together, like coordinated dance, and all it takes is one thing to trip or mess up, and the whole thing goes haywire. There's so much things that could go wrong that frankly it's a wonder that we all end up here in one piece. Co-dominance is believed to be the violation of the law of dominance, when the alleles for the particular trait are co-dominant. They are both expressed equally rather than a dominant allele taking complete control over a recessive allele. This means that when an organism has two different alleles, it will express both at the same time. In complete dominance, sometimes in heterozygote dominant alleles does not completely mask the phenotypic expression of recessive allele, and there occurs an intermediate phenotype in the heterozygote. With codominant alleles, both traits are expressed at the same time. With incomplete dominance, the same occur but the traits are blended together, rather than occurring in discrete patches. It thus refers to the condition in heterozygotes, where the phenotype is intermediate between two homozygotes. Polygenic inheritance occurs when one characteristic is controlled by two or more genes. Some traits are controlled by many genes such as height, IQ, skin color, eye color, etc. Often, the genes are large in quantity but small in effect. In 1913 Davenport, Jamaica found that two pairs of genes, A and B, caused the difference in skin pigmentation between Negro and Caucasian people. These genes were found to affect the character in additive fashion. Multiple alleles, Mendel studied just two alleles of his P genes, but real populations often have multiple alleles of a given gene. Pleiotropy. Some genes affect many different characteristics, not just a single characteristic. Gene linkage is the type of coexistence of two or more genes in the same chromosome. If a genes are situated in the same chromosome and are fairly close to each other, they tend to inherited together. Each chromosome contains more than one gene. Extranuclear inheritance, though, the genes of nuclear chromosomes have a significant and key role in inheritance of almost all traits from generations to generations. They altogether cannot be considered as the sole vehicles of inheritance.